Today we're going to speak about, we're going to discuss evolution and immunity. First I'm going to talk about why immunology is important in medicine. I'll briefly describe a little bit about some of the cells involved in the immune system and the immune response, and then we'll talk a bit about the anatomy of the immune system. And with that background, we're going to talk about the functioning of the immune system in two contexts. One is how the lymphoid system develops. That goes by the name ontogeny as opposed to phylogeny, which is evolution over long time scales. That's what phylogeny is. Ontogeny is evolution over shorter time scales on the scale of the lifespan of an individual organism, such as a human being. And then lastly, the final section is going to be about the phylogeny of the immune system. And all of this is going to try to drive home the take-home lesson, which is that the immune system evolves on 2 or maybe 3 time scales, depending on how you want to count it. And so the essence of what I want to get across is that evolution isn't just sort of a peripheral aspect of immunology and immunity and the immune system and how it functions. It's absolutely central. So I want to share with you just one sort of liberal arts allusion. It's allusion, not illusion, which is a quote from the Peloponnesian War, a famous book by the famous ancient Greek historian Thucydides. And what it describes was an epidemic in Athens during the period of the war with Sparta. Unfortunately, we're the Spartans, so I guess that would make us opposed to the Athenians. It was with those who had recovered from the disease that the sick and the dying found most compassion. These knew what it was from experience and had now no fear for themselves, for the same man, presumably or woman, was never attacked twice, never at least fatally. So this is already a description which is probably relevant to immunology. It's probably describing the phenomenon of immunological memory, whereby exposure to a foreign microorganism, a pathogen that causes disease, leads to better resistance. And that's sort of a hallmark of what we call adaptive immunity. Now, why is the immune system relevant? Well, I've already mentioned development. So like the rest of the body's systems, like your kidneys or your liver or your blood-forming cells, the immune system develops from the stage of the embryo and the fetus through early life, neonatal life, and then into childhood and into adulthood. Physiology, the immune system not only deals with immunity to foreign microorganisms and sometimes macroorganisms like worms, which I'll briefly mention later, but it's part of our normal physiology. The immune system is tied into all sorts of other processes that are part of the normal functioning of the body. Things like wound healing and hemostasis. There are influences between these other systems and the immune system. Pathogenesis of disease. The processes by which diseases develop over time often involve the immune system in multiple respects. Diagnosis. We use products of the immune system, most notably antibodies, which I'll be talking about a bit later, to help diagnose diseases, to assess the nature of a disease, to make prognoses in some cases. They're used every day in the clinical labs across the street at university hospitals. Prognosis I just mentioned as part of diagnosis, and therapy. There are more and more examples of products of the immune system being used to treat patients with all sorts of diseases, from lymphomas and leukemias to autoimmune diseases. Some of you may know people who've been treated with monoclonal antibodies, a particular kind of antibody that's extremely useful both in the laboratory and in the clinic at the bedside. And then prevention. Probably the single most important of immunological understanding and the technology involved with immunology is the development of vaccines which have saved countless millions of people. And here's an example that shows you that this is just 3 different vaccines. Diphtheria, which is probably the oldest that's been in routine use for many decades, polio, and measles. And you can see in each case where time is on the x-axis and the number of cases reported is on the y-axis, that it's like falling off a cliff. So here's the incidence of diphtheria up to the introduction of the vaccine, and then look at that fall of incidence. Absolutely spectacular. Look at polio. Look at measles. There was one problem where they changed the vaccine and used a killed vaccine, and it had some unanticipated consequences causing a disease. But in general, the incidence of measles itself dropped off drastically. And this is why it's so important to keep vaccinating people, and why it's problematic when parents decide on their own not to get their children vaccinated, because it decreases what's called herd immunity and increases the likelihood of greater incidence. The single greatest success for vaccines is undoubtedly smallpox. And as many of you may have heard, Edward Jenner in England in the late 1700s was responsible for the first publication. Others had preceded Jenner in what we now call variolation, in using a smallpox-related virus called cowpox to inoculate individuals and protect them against smallpox, which was much more virulent. So he started the ball rolling, but when we finally had a modern smallpox vaccine, a worldwide effort organized through the World Health Organization eventually was able to declare smallpox eradicated in 1979. And that is certainly the greatest achievement so far. Now let's talk about the relevance of the immune system to pathogenesis. So what are the kinds of diseases where immunity and the immune response can be involved? Well, we have infectious diseases. That seems obvious. In fact, the protection we have against most of the diseases for which we vaccinate is based on the induction of an immune response, generally, most notably an antibody response. That's one kind of immune response, but it generally involves activating other arms of the immune response as well. So whether it's a bacterial infection like pneumococcal pneumonia, or a virus like influenza or HIV, or a fungal infection or a parasite infection like malaria, the immune system is virtually always involved. We have allergic diseases or hypersensitivity diseases. Many of you are probably sufferers of hay fever or some other allergy. Generally about 1 in 10 or so people have some sort of allergic symptomatology. Autoimmune diseases like multiple sclerosis or systemic lupus erythematosus or rheumatoid arthritis. Many, many more examples. Immunodeficiency diseases. Now these are less common when they're very drastic, and this is mostly a problem for pediatrics. But as our technology has advanced and as we've become more capable of finding immune deficiencies, subtler immune deficiencies, we find that more and more people have some degree of what you can call a functional deficit, even if it's a very narrow one that only predisposes to one or a few kinds of infections. Cancer. There are cancers of the immune system, like leukemias and lymphomas. But there are also other cancers for which the immune response is important. And also, the immune system can be exploited for therapy as well as diagnosis. It's very widely used for diagnosis, but also for therapy, in some cases, highly effective therapy. And diseases that result in organ failure, because then we do transplants in many cases, particularly for kidneys, livers, hearts, lungs, and occasionally other organs. And there, regulating the immune response, the immune system is crucial. Here's an example of an excessive immune response to what is actually not a threat to the body. It's poison ivy, and that results from a chemical in the plant which functions as what we call an antigen, a foreign substance that elicits an immune response. This is based on what's called a cell-mediated immune response, not antibodies, but a cell-mediated immune response. And this is the eruption in the skin there is all due to the immune system. Now, I'm going to briefly describe some of the terminology in the immune system, some of the cells that are involved, and I hope you won't be overwhelmed. There are a lot of different cells. We're not going to talk about all of them. We're going to focus on only 2 tonight, really. All the cells we're going to focus on tonight are white blood cells. So in the blood, we have white blood cells, and then we have several kinds of white blood cells. We have lymphocytes, and we're going to focus on B and T lymphocytes primarily. The B lymphocytes make the antibodies, and the T lymphocytes help the B cells make antibodies and also have other functions. They also secrete soluble molecules, as do the B cells, that are called cytokines. These are just messenger molecules. They're secreted by one cell, and they tell another cell what to do, or even sometimes the same cell. There are what are called natural killer cells and dendritic cells, and we're not going to get into their functions in any detail this evening. Then there are what are called the granulocytes, the neutrophils, basophils, and eosinophils. The neutrophils are the most common white blood cell in the blood. And they are the first to arrive at most infections or most cases of inflammation in the body. And then there are monocytes, which when they get into the tissues are called macrophages. Now, in the blood, there are also other cell types called erythrocytes or red blood cells. Those are the cells that carry the hemoglobin we talked about in the context of sickle cell disease last week. Everybody's probably heard of red blood cells. If you go to the doctor and you're anemic, it's because you have too few red blood cells. And then platelets, which are involved in clotting, hemostasis. So if you have a cut, the platelets help plug up that injury so you don't bleed excessively. Now, it turns out these cell types are also involved in various immune phenomena, but they're not generally considered core members of the immune system. The core members of the immune system are the lymphocytes and these other cells, the white blood cells in the blood. But there are lots of other cell types in the tissues and even the parenchymal cells, the cells that make up organs like liver cells and kidney cells. They actually play a role. So the concept of the immune system as a totally separate entity is a simplification. It's a useful simplification, but a simplification nonetheless. So here's a schematic view of all these cell types. And we're going to focus on the cells over here on the left in blue. Those are the lymphocytes. That's why they're separated out by color. And we're going to focus on the B cells and the T cells. And the B cells are the ones that make antibodies. And the T cells, as I said, in some cases, there are different kinds of T cells. Some of them help B cells make antibody, and some of them have other jobs in the immune response. And the way this schematic works is they all arise in the bone marrow. Then they go into the blood, and then some of them go into the tissues, either lymphoid tissues, like the lymph nodes. You've all heard of lymph nodes. When the doctor feels under here, if you come in because you don't feel well, you might have an infection, he's feeling for enlarged lymph nodes. Because when you get sick and there's an immune response, your lymph nodes get bigger because there are more cells, because there's cellular proliferation. In other words, the cells are reproducing. They're making more of themselves to respond to the foreign invader. There are these cells that have an interesting shape called dendritic cells. And again, these were the focus of a Nobel Prize this past fall. And then there are cells in the tissues like macrophages and mast cells. Mast cells you may have heard of if you have allergies. And then there's the final effector stage of these cells. The ultimate end of the B cell lineage, as it's called, is the plasma cell, which is specialized to make antibodies at a furious pace. Now, this is the anatomy of the immune system at a very low resolution. So there are what are called primary lymphoid organs, the thymus and the bone marrow. All of the cells ultimately come from the bone marrow in adult life. But the T cells first have to go through the thymus. So the thymus is like a school for T cells. And we even use the terminology that T cells are educated in the thymus. The rest of the cells come from the bone marrow. But there are also secondary lymphoid tissues like the spleen, which is the largest secondary lymphoid tissue or organ, and then lymph nodes scattered all through the body. And there's also a separate vascular system. In addition to the blood, the veins and the arteries, there's the lymph system. And we have two basic flavors of immunity. Innate immunity, which is less discriminating, not really enhanced by prior exposure, and has a relatively rapid response in the order of minutes to hours to begin a response. Whereas adaptive immunity, which involves the B cells and the T cells, has a vast range of molecular discrimination. You can tell one flu virus from another, or one virus from another virus, one bacterium from another bacterium. It's enhanced by prior exposure, and that gets back to that concept of memory I mentioned in the quote from Thucydides. And it's relatively slow. It takes days to weeks. And the reason it takes so long is it requires the cells to proliferate. And our cells take a day or so to duplicate themselves, or at least 12 hours. And then there's a difference between systemic immunity in your blood and your organs and your mucosal immunity, which is the linings, the epithelial linings of your pulmonary system, your urogenital tract, your gastrointestinal tract. Now, lymphocytes circulate around the body, both in the blood and the lymph. And this is a very schematized view showing you that they go around in the blood, in the veins, and then the heart, and then back with the arteries. And then they go through the secondary lymphoid tissues like a lymph node, and that involves also transiting through the lymph vessels. And if we put this rather schematized view back into the body, you can see that the largest lymph vessel is called the thoracic duct. It empties into the left subclavian vein. So the lymphocytes are circulating around in the blood and sometimes in the lymph. And here's just showing you that if you don't have proper lymph drainage, this is what can result. This is called elephantiasis. It's due to a worm infection. So we're going to stop now and take some questions and then move on.
I'd like to talk about the immunization program for children. Why don't we do it as adults? Because after a while, they say you have to get re-immunized.
Well, because there are certain diseases that children are highly susceptible to. And some of these diseases are really extremely dangerous, like measles, which can be lethal in some cases or cause brain damage. So there are a lot of diseases which young children are susceptible to a much greater degree than adults or even older children, particularly with certain kinds of bacterial pathogens. There's actually, I think, very strong rationale for immunizing young children.
Children and not adults.
Well, you are supposed to get boosters for certain kinds of vaccines. I don't know that most people actually do that, but I think we still have to immunize young children.
What is the role of stress in the pathogenesis or molecular and morphological changes and structural alterations?
Well, I'm not going to get into that tonight, but stress has a very significant impact on immune system function. And some of the hormones and mediators that are involved in the stress response, like glucocorticoids from the adrenal gland and noradrenaline or norepinephrine, have significant impact on the functioning of lymphocytes. So it's generally the case that chronic stress will degrade immune system function and lead to greater susceptibility to infection. That's a short answer.
I would think that most organisms function to better themselves. So, for example, if something invades your body, it's trying to multiply to create more of itself, and your body's a good host. But why would the body attack itself in autoimmune situations? I mean, that seems contraindicated to being good for the organism.
I can't get into anything close to the level of complexity which would provide a really satisfactory answer. But the short answer is the immune system involves an enormous number of very complex interactions among cells and various kinds of molecules, and it requires a great deal of regulation. And there are all sorts of genetic variants which will influence that regulation. And going back to what we talked about last time, this concept of antagonistic pleiotropy and the fact that there are often trade-offs, genotypes and phenotypes that are advantageous in one set of circumstances may in fact be disadvantageous in another. So for instance, there are both allergies or hypersensitivity diseases and autoimmune diseases are basically both examples of excessive immune responses or poorly regulated immune responses. And it turns out there's some evidence that people who are more susceptible to allergic diseases are more susceptible in part because they have more of a certain kind of response which makes them better protected against certain kinds of parasitic worms. So that's the kind of thinking that you need to engage in. In other words, that evolution frequently requires trade-offs. So there is no perfect genotype or phenotype. The environment, particularly as it changes, can cause a genotype that's great in one circumstance to not be so great in another.
I believe that I recall there was an era when it was thought the thymus was not necessary and it was surgically removed in children, and I just wondered what effects were seen.
That was wrong. Not a good idea, and I don't know specifically what sort of deficits those people might have had, but they used to irradiate the thymus, actually, aside from surgical excision, and in some cases they got tumors, I think. But it wasn't a good practice, and fortunately that was stopped. The key point here is that lymphocytes recirculate because the immune system has to carry out what's called immune surveillance. It has to keep track of what's going on everywhere in the body, in every tissue, because almost any cell type in your body is susceptible to infection by one kind of microorganism or another. So this slide is just meant to indicate that the mucosal immune system is also important. It's not just the systemic immune system that matters. And many of the infections you get, if you think about it, come through your mucosal tissues. In other words, you breathe them in, or they come in through eating or through some other exchange with the environment, shall we say. So what this slide is meant to illustrate is that there are a variety of mucosal tissues in the gut, the urogenital tract, and the pulmonary system. And the lymphocytes circulate in the blood, and they also collect in the lymph and then get back into the blood, coming out of the mucosal system. And then effector cells, if you have a stimulus to the immune system, what we call an antigen or an immunogen, they will then distribute throughout the mucosal tissues. So I don't want to dwell on that. I just want to make you aware that there's a very important role for the lymphocytes involved in protecting the mucosal surfaces. So now I want to get into how the immune system actually functions. And I have two little sections here, or three, depending on how you count it. The first two slides are about the development of the immune system. And I'm specifically focusing on T cells, which, as I mentioned, are educated in the thymus. And what I mean by educated is it's a little bit like Goldilocks. You want your T cells to have a receptor, each different T cell to have a receptor that recognizes one or a very few foreign molecules from this virus or that bacterium or this parasite. And you want it to recognize it well, but not too well, because it turns out, and this is based on a discovery that won a Nobel Prize, your T cells, unlike your antibodies, simultaneously recognize a self molecule when they recognize a foreign molecule. This is somewhat counterintuitive, and that's why it got a Nobel Prize. So in the thymus, there's two kinds of selection. There's selection for cells to recognize self, and this is a kind of cell we call an epithelial cell that exists in the thymus. It's not from the bone marrow. And here we have a T cell. And on the surface of that epithelial cell is a molecule we refer to as an MHC molecule. And there's two kinds, and we're not going to get into the details. All you have to know is that this kind of molecule is on the surface of that cell, and the receptor on the T cell can bind to it or not to varying degrees. And if it doesn't bind at all, it dies. It commits suicide. It's what we call programmed cell death, or apoptosis. If it binds well enough, in other words, it's sort of like Goldilocks, then it survives and it continues developing. And this goes on in the part of the thymus called the cortex. It then goes on to the medulla, where there's a whole bunch of other cells, not epithelial cells, generally dendritic cells or macrophages. These are other non-lymphocytes that also can express this MHC molecule. And in this context, if it binds too tightly, it dies through programmed cell death. And if it binds moderately, then it lives. So what you want is self-recognition that's not too weak and not too strong. It has to be just right. But the key point I want you to take away is that this is a selective process that is superimposed on a random process of generating these receptors. Each of these receptors has a unique amino acid sequence. It's just a couple of polypeptide chains. And each unique receptor has a different amino acid sequence. And that sequence diversity is generated at random. The body doesn't know which receptors are going to come up, and how it determines which ones to keep and which ones to throw away is by this selection process. There's no central regulator determining who will live and who will not. And in fact, more than 90% of the T cells die in the thymus. Now that may sound wasteful, but it's the only way biology could come up with to achieve the end of a suitably sculpted repertoire, that's the word we use, for the entire library of T cells and their T cell receptors. Now let's talk about an immune response. Now this is a simple way of looking at immune responses. We can talk about antigens as molecules that, when they're exposed to an antibody or a T cell receptor, can form a physical complex where the two actually stick together, sort of like two pieces of a jigsaw puzzle. And that's a biochemical event. But the more relevant biological event, which it often results from this biochemical event, is what we call an immune response. And that involves an antigen in the form of what we term an immunogen. If you take an antigen, inject it into an animal, or somehow get it into an animal, like having the animal inhale it, or by injecting it, or eat it, then it can come into contact with lymphocytes, B cells and T cells. And the end result is a process by which these cells proliferate, meaning they make more of themselves, and differentiate. They change what genes are being expressed, acquire new functional capacities, and that is what an immune response is. And they generate what we call effector cells and memory cells. And the memory cells are partly what accounts for that observation of Thucydides from a couple thousand years ago. So this is what it looks like with an electron microscope. This is a small resting lymphocyte, and when it is exposed to its antigen, it doesn't matter if it's a B cell or T cell, it'll look pretty similar, even though these are distinguishable cells by techniques we now have. But looking through an electron microscopic image, you don't really see a difference between a B cell and a T cell, so this applies to either. But when you activate a small resting lymphocyte, you get an activated B cell, if it's a B cell, or an activated T cell, an effector cell. And these are the cells that actually cause the mediation, or that actually mediate immunity. So that's, at its simplest, that's what an immune response is. It's taking a small resting cell, what we call a quiescent cell, activating it. And what that means is that it changes its gene expression pattern. It turns off some genes, it turns on other genes. It might start secreting cytokines. It might put new molecules on its surface. It might now be able to crawl around better and get into tissues better, and so forth. And the end result is an effector B cell in the form of a plasma cell, which secretes lots of antibodies, or an effector T cell, which can either secrete cytokines or kill other cells, a so-called cytotoxic T cell. Now, I'm not going to go into any details beyond that, except to talk about the clonal selection concept. So when you have a group of lymphocytes that all have the same receptor with the same amino acid sequence and that recognize the same antigen or antigens, we call that a clone. So it's a group of cells that are all derived from the same precursor cell and are genetically identical. And this concept of clonal selection was the basis for a book by an Australian by the name of Macfarlane Burnet, who went on to win a Nobel Prize for this idea. And this is the original schematic from his book, which led to a Nobel Prize. And this is called the clonal selection theory of acquired immunity. And the basic idea is each of these little circles here with a letter in it represents a different clone. And the different letters correspond to different receptors, antigen-specific receptors. In this case, he was talking about B cells because we didn't know very much about T cells in 1959. So an A is one sequence of amino acids for that antibody on the surface of that B cell. And B is another amino acid sequence. And each of these different amino acid sequences of these receptors, as illustrated by the schematic antibody here, recognizes a different antigen. Now, if the antigen for clone C comes into the body, then that antigen confers on clone C a selective advantage. So all the lymphocytes with that clone C receptor will reproduce at a faster rate than clone B or clone D. The net result is over time, the distribution of clones in the population has changed. That is evolution. It's evolution on a cellular level as opposed to an organismic level. This is ontogenetic evolution, or maybe subontogenetic evolution. This happens in the course of an individual immune response. Your immune system is literally evolving every time you have an infection. Now here's a more schematic, I shouldn't say more schematic, it's equally schematic, but a more colorful way of illustrating these same points. So this is an overview slide, and I will now break this down into a little bit easier to see segments. So you start out with an initial repertoire of lymphocytes that are generated. Again, the receptors are generated by a fascinating process, which I am not going to get into because it's very complex and involves a lot of DNA biochemistry. But it's a process where you randomly create sequence diversity in these receptors. That means different amino acid sequences, different strings of amino acids. And each cell has a unique receptor. What I showed you in the thymus for T cells, and a comparable process occurs for B cells, but not in the thymus, in the bone marrow, is if you have self-recognition that's too strong, you're deleted. We don't want a lot of T cells recognizing, or B cells recognizing self-components causing autoimmune disease. And actually getting back to the question, there's actually a gene in the thymus that's very crucial for the education process of T cells. If you have a defect in that gene, you end up with an autoimmune disease. So there's one example where if this process of deleting self-reactive clones is defective or inefficient compared to average, you can end up with autoimmune disease. So you shape the repertoire, you sculpt the repertoire by deleting those elements of it which potentially will cause autoimmunity. At the next stage, once you've gotten rid of the self-reactive clones of lymphocytes, you then, and all that happens without any antigen being introduced into the system. Totally antigen independent. Once you're exposed to a foreign antigen, like this year's influenza virus, or a pneumococcus, or E. coli from the wrong hamburger, then the lymphocytes with the right receptors become activated, and therefore they reproduce more quickly, and the end result is antibodies. And so what you end up with is the greater numbers of lymphocytes that represent the clones that are specifically the ones able to deal with that antigen. It's actually a brilliant solution to a very difficult problem. How do you react to effectively an unlimited universe of antigens? You can't know ahead of time what you will encounter. And these are just the ideas of the clonal selection idea. I'm not going to go through them. I think I'd be happy to do it in the question and answer session out in the atrium, because it'll just take too long. But I hope you have the basic idea that this is a selective process, and the lymphoid population is evolving in real time in the course of an individual immune response. And it's also evolving in the course of your development from fetal neonatal life to adulthood. Now let's talk just a moment about antibodies, and then we'll move on to the final section after a question and answer period. Antibodies are schematically illustrated here. They're made of two kinds of polypeptide chains. Two identical heavy chains, which would be these two long chains, and two identical light chains. So the heavy chains are in green, the light chains are in yellow. Each of these chains has two kinds of domains. Sorry about the terminology, it's just the complexity that's out there. There are variable domains and constant domains. And as the names would imply, variable domains are more variable in amino acid sequence. Constant domains are more conserved in amino acid sequence from one antibody to another. This is a T cell receptor, which is very similar in structure to an antibody, but it's sort of a smaller version of it. Now, antibodies can be on the cell surface of B cells, and they are on the cell surface in terms of recognizing antigen initially. But then they get secreted. T cell receptors don't ever get secreted. But antibodies get secreted and do their work in the body fluids, in the blood and the tissue fluids. Okay, so these are the molecules that actually mediate immunity, what we call humoral immunity. And again, this focuses in the red domains are the variable domains of the heavy chain and the light chain. They are the domains that recognize the antigen. So if you have an antibody against influenza virus, that's because its VH and VL domains together form a pocket that specifically can fit onto a component of that virus. And there's a whole set of mechanisms that diversify antibodies. We don't have gene conversion in our genomes, in our B cells. That happens in some other species, other mammals and birds. But we do have somatic hypermutation and class switching. I'm not going to talk about class switching. I just want to focus for a minute on this concept. Antibodies are unique in that, unlike any other genes in the body, they undergo a process of accelerated mutation that is localized specifically to those variable domains. And on the next slide, here we have an example of what we call somatic hypermutation with affinity maturation. Now let's break that down. Somatic hypermutation just means that it's a process that goes on in the somatic cells as opposed to the germ cells. It's not in your gonads. It's in your B cells out in the tissues or in the blood. And it's a rapid mutation process, hypermutation. That means it's faster than normal. Affinity maturation means that as you accumulate mutations, the affinity of those antibodies for their antigens, their ability, the strength with which they bind those antigens goes up. And what you're seeing here is that over the course of an immune response, day 7 after immunization, day 14, day 21, each of these lines represents an amino acid sequence, and each time there's a red bar, that's a mutation. And what you can see is over time, there are more and more mutations in those sequences. And I'll give you a real-world example. People are very interested now in creating a vaccine to HIV. And it turns out that the antibodies, the only antibodies that have the ability to neutralize, to inactivate HIV viruses of any origin, what they call potent, broadly neutralizing antibodies. Those antibodies tend to have enormous numbers of somatic mutations. So in other words, in order to combat HIV ideally, you need to have very substantial evolution of your antibodies. So in order to fight the evolution of HIV, which is the most variable virus we know of, maybe with one exception, certainly one of the most variable, we need to have a similar process in our own immune system.
Is it possible to remove T cells, grow them in vitro, maturate them, and then inject them back?
In fact, people are doing that as we speak. So the question is, can you take T cells out of the body, mature them, activate them, grow them in vitro, meaning in a lab, in tissue culture, and then put them back into the individual? People are doing what they call adoptive T cell immunotherapy. And in fact, my first student is now at MD Anderson. My first graduate student, PhD student, he's actually an MD PhD, and he is doing exactly that kind of work. They genetically engineer T cells. They take T cells out of your body, they genetically manipulate them to make them able to recognize a particular tumor, or they can do it potentially for a specific virus, and then reinfuse them. And in some cases, there was actually a paper from the New England Journal of Medicine, in the New England Journal of Medicine, just recently in the past 4 to 5 months, I'd say, from a group at Penn doing something along that line. And they had some very promising initial responses of some cancer patients. So that is doable, and the technology exists, and it's being used.
What is T-cell lymphoma?
So T-cell lymphoma is a cancer of the T-cell system, and it involves some sorts of genetic mutations that make those T-cells dysregulated, and they grow without regard to the normal constraints. And lymphomas generally mean that it's a collection of cells, tumor cells, in a localized place in the tissue, like a lymph node or a collection of cells in a tissue, as opposed to a leukemia, which is in the blood. So it's a form of cancer. It's a malignancy of the T cells. There are also some viral causes. It's either a virus or it's due to a genetic mutation or a set of genetic mutations. Those are the main 2 ways you get a lymphoma or a leukemia.
I wondered if someone found out, recently that they had cancer and they wanted to have your graduate student help him. Is that possible?
I don't, off the top of my head, I don't know what criteria they're using, but I can certainly provide the name of the group at Penn, the main person, and the name of my former student, and you can contact them and see if it's the right kind of cancer and if they have the right clinical situation to try that kind of therapy.
Somebody said in an earlier lecture that the reason why there's no vaccine for HIV is that it mutates so fast that by the time you got a vaccine, it would have changed. Is that correct?
That's one of the reasons. So the question is about why is there no HIV vaccine yet? And there are a number of reasons. First of all, it's been very difficult to determine exactly what would be the immune mechanism that mediates immunity. So if you look at bacteria like Streptococcus pneumoniae or pneumococcus, we know exactly what sorts of immune mechanisms actually mediate immunity. In that case, it's antibodies to the polysaccharide capsule. That's the key mechanism, and we can make vaccines which elicit those kind of antibodies, and we have a successful, in fact, we have 2 successful vaccines for pneumococcus. For HIV, we're still figuring out what is the best immunological mechanism or mechanisms to mediate immunity, And then there's the problem that HIV mutates enormously rapidly. And I'll just give one little point and then go back to the lecture, which is a little while ago, I came across a paper that pointed out that if you look at the amount of diversity of HIV in a single patient, it is on the scale of or greater than the entire worldwide diversity of influenza virus. And I was staggered by that, because influenza virus is among the more variable viruses. And we generally think of it as mutating and changing pretty rapidly. And there's more diversity in a single patient with HIV infection than all the influenza virus diversity in the world. I'm just amazed by that. So I think it is a truly amazing challenge. And essentially what that means is then it's difficult to make a vaccine against a pathogen that's evolving at that pace. So this last section, which will be a little lighter, I hope, for you, although the terminology is still an issue, I'm sure, is going to be about phylogenetic origins of the immune system. And again, this is going to be a very brief and a very high, I should say, low resolution from high above view of the evolution of the immune system. There's way too much complexity to get into details. This is what we call a phylogeny or a cladogram, which illustrates the evolutionary relationships, the relationships of descent of humans to a lot of other species or types of organisms, including plants. So the last common ancestor to plants and all these other animals is the farthest one back. And as I mentioned last week, we are eukaryotes. All animals, all plants, and all fungi are eukaryotes. Then we have bacteria and archaea, and those are the other domains of life. So we are more closely related to an oak tree than we are to a bacterium, according to this kind of thinking. So this just gives you a sense of the scale. So I mean, if you go back to insects, we're talking our last common ancestor with insects was hundreds of millions of years ago. Last common ancestor with reptiles and birds, tens of millions of years ago, maybe hundreds. So anyway, this just gives you a sense of the scale. So now, instead of talking about evolution of individual cell populations in a given person on the order of days and weeks, or hours even, we're talking about evolution over millions of years, tens and hundreds of millions of years. Now, just to give you a sense, and this would be true for most systems in the body, of how much conservation there is among the species, going back way into the distant past, evolutionarily speaking. These are molecules called defensins. These are small peptides which mediate part of what we call innate immunity because they don't require lymphocytes. They can directly kill bacteria. And we have these peptides in our skin, in our guts, and various other places, in our mouths. And this is a similar peptide from a radish. And this is a three-dimensional structure based on a technique called X-ray crystallography. You shoot X-rays at a protein crystal, and the diffraction pattern allows people with the proper intellects, mathematically gifted people like Glenn and Dan and Cyrus over here, to figure out the distribution in three-dimensional space of all the atoms in that molecule, which is amazing because a protein has many hundreds or thousands of atoms. And so these are sort of representations of the backbone of the polypeptide chain. And there's different kinds of what we call secondary structures, like an alpha helix or a beta strand. And what you can see is they look enormously similar. And we're talking about many hundreds of millions of years of evolution distinguishing radish from a fruit fly from a human. But here is an example of a highly conserved structure because it's effective. It's selected for. There's clearly some variation, but you can see that the basic structural motif is enormously conserved. Similarly, if we look at what's called a signal transduction pathway, this is a series, it's sort of like a Rube Goldberg mechanism on a molecular scale, where something binds to the receptor on the surface of a cell, and that then causes that molecule on the surface of the cell to interact with another molecule or a bunch of molecules, and so on down the line, until you send a molecule into the nucleus to bind somewhere on a chromosome and turn on a gene or turn off a gene. That's what we call signaling or signal transduction. Here are two signal transduction pathways, one from the fruit fly and one from us or a mouse And this is called the toll pathway. And again, this was also the basis for one of the Nobel Prizes this past October for Bruce Beutler. And you can see again that there's an enormous amount of similarity. Now, some of that is just the artist drawing the little shapes the same. Each one, this is a molecule called toll-like receptor. There's several of them in humans, up to 10. And this is a molecule called toll, which is German. I forgot what toll means in German, but that's how it was named. And originally, this molecule was believed to be involved in the development of the fruit fly. That was its function. And it is. It's just, it turns out it has other functions in immunity to fungi. And there's a whole set of toll-like receptors where the name TLR comes from, toll-like receptor, in humans that have to do with recognizing that we have been invaded by bacteria or by viruses or by other kinds of foreign entities that we're not happy to be invaded by. And again, there's enormous conservation of the basic steps in the pathway. So the point, again, is that when a system evolves, although it will change in various ways, it will be tweaked by mutations, because mutations just happen. But selection will, if it's functional, selection will maintain many of those structural features because they determine the useful functions. Now, here is a rather imposing table which shows you for a variety of molecular mechanisms involved in immunity. And here across the top are all the different species. We have insects represented by Drosophila, sea urchin to represent echinoderms, sea squirts for ascidians, lamprey, which are jawless vertebrates. There are only 2 jawless vertebrates, if you didn't know, lamprey and hagfish. Then we get into the vertebrates: sharks, cartilaginous fish, carp, which are bony fish, amphibians like frogs, reptiles like snakes, I think that says snake, birds like chickens, and humans. And what you can see just by the no's are in white and the yeses are in red and the unsure are in pink, what you can see is beginning with the jawless vertebrates, you begin to see an enormous amount of similarity in immune mechanisms. These are different sets of molecules that are involved in immunity that are conserved across all these species going back to the jawless vertebrates, which is going back a long, long way. And included here, we have all the elements of adaptive immunity, like immunoglobulins, T cells, MHC molecules, which I mentioned briefly, other molecules which I didn't get into called complement, mannose binding lectin, et cetera, toll-like receptors. See how ancient toll-like receptors are? They go all the way back to fruit flies and probably even a little further back. So the basic idea then again is that our immune systems share an enormous amount with mice and even worms and fruit flies. And that's why it's, although Sarah Palin may not think so, most scientists recognize that studying fruit flies and studying worms and studying the zebrafish, which is a very recently developed model for all kinds of biological studies, is actually useful. And even though Drosophila don't get the exact same diseases we do, or worms, or even yeast, it turns out that there's enough conservation of fundamental molecular level and cellular level mechanisms in these ancient, evolutionarily ancient species to inform our own investigations of the human physiological systems, whether it's the immune system or the blood clotting or system or the kidney. And obviously, you can't study kidneys and yeast, but it's an amazing and very valuable property of the living world that we can learn a great deal about our own behavior from a scientific point of view by studying what goes on in other species, which are easier to manipulate, and it's more ethical to manipulate them in certain ways, which none of us would want to submit to. So this next slide just illustrates what I want to leave you with, which is this notion of theme and variation. I don't want to go into the details. That's not the point. What you can apprehend from this slide is just look at the colored patterns here. Each one of these rows represents a certain portion of the chromosome where you have a rearranging antibody locus. You have a whole bunch of gene segments, and the antibody genes actually jump around a little bit. There's a whole process called rearrangement that is involved in making complete antibody genes that encode the complete antibody proteins I talked about. And the only point to take away is that you see that each of these kinds of genetic elements is color-coded. Doesn't matter what they are. Just notice that they all have what are called V domains. They all have Ds, or most of them have Ds and Cs and so forth. The whole point is that each species, each one of these lines is a different species. Each species has a somewhat different arrangement. And this is generally how evolution works. There's a lot of similarity. There's a lot of conservation, as we use that term to mean sameness. But there's also, some variation in terms of the number of genetic elements or the exact relationship to one another in one dimension, and exactly what process ensues. But the main point to take away is that they encode a bunch of different kinds of antibodies, all of which have the same basic structural motifs, all of which have the same basic functional properties of being able to bind antigens. And when they bind antigens, they then also can activate lots of antigen non-specific mechanisms that can destroy that antigen or clear it from the body. And so a major theme in the immune system is linking functionally and physically recognition of a threat with the means to destroy that threat. And generally, the means of destroying it are not specific for that antigen. The antibody is specific for the antigen, but the systems it activates after binding the antigen are not specific for the antigen. They'll destroy anything. And that's why if the regulation of one of these mechanisms is defective, you can destroy your own tissues, because the destruction mechanisms are very nonspecific. They can't tell if they're destroying a self cell or a yeast cell, a self cell or a bacterium. Now, sometimes we actually do destroy our own cells on purpose. Like when we have a viral infection, our T cells can actually destroy the cell before all the viruses get released, thereby preventing the reproduction of the virus to the same degree. So let's conclude. Immune function is relevant to medicine in multiple ways, including, as I illustrated, pathogenesis, diagnosis, therapy. There are many other possibilities. Immune functions are mediated by a variety of lymphoid and non-lymphoid cells. I focused on the lymphoid cells tonight, and that's a chauvinistic aspect of immunologists. Many of them tend to focus on lymphocytes, but we all recognize, or at least most of us do, that these other cell types are also incredibly important. And it's really about the interactions between them. The immune system includes tissues distributed throughout the body, and mobile cells visit these distributed sites. It has to be that way. Otherwise, how would you know if you had an infection in your foot or an infection in your hand or your lungs? You have to have cells constantly traveling around. And remember, there's only a tiny minority of cells which have any one specificity. So they have to constantly be searching around looking for the antigens to which they can respond, because other clones won't be of any use to that antigen. And immune cell development and the functioning of the immune system, immune responses, involve selection and evolution of cell populations. So evolution is very much built right into the functioning of the immune response. Immune responses, as I just noted a moment ago, link antigen-specific recognition, antigen-specific receptors, to antigen-non-specific mechanisms of antigen clearance or destruction. Adaptive immune responses involve clonal competition and evolution, for which Burnett got his Nobel Prize. And immunoglobulin genes are subjected to a unique hypermutation mechanism that facilitates the evolution of better adapted antibodies. Now, I didn't mention, but I will now, that not every mutation makes the antibody better. Most of them make them worse. And what happens to the cells that have a worse receptor is they die. So once again, there's what appears from our perspective to be profligate waste. Nevertheless, that is the only way we can deal with the fundamental challenge the immune system faces, which is to be able to respond to any of potentially billions of different threats in terms of molecular structure, without knowing ahead of time which ones we'll be exposed to. And many aspects of the immune system function, arose hundreds of millions of years ago in ancestral species. And although those mechanisms have been tweaked and are continuing to be modified and adjusted according to our circumstances as we are now, it's because of this enormous conservation of molecular detail and cellular detail that we can study other species and gain information of great value to the diagnosis and treatment of disease in human beings. And finally, there are many variations on fundamental structural themes of immune system molecules among different species. So I guess I already said that. I'll stop there, and we'll have questions after dinner. Thank you.